Hi guys, welcome to our YouTube channel Gamsapi. This was hosted by the Gampa Medical Students Association, including me and my colleagues who have qualified to enter the medical faculties of Sri Lanka after doing the A-levels. And the main reason that we decided to start this YouTube channel was to help you guys to face your exam successfully at this moment of crisis, the COVID-19 pandemic that we all are facing, and you guys are unable to attend your classes to complete your syllabus. So this video will be on Unit 9, Microbiology. And this video will be on the nature, distribution, and the role of soil microorganisms. So starting off, soil provides an adequate physical and chemical environment for growth of microorganisms in terms of space and nutrients, which include minerals, decomposing organic materials, water, gases such as carbon dioxide, oxygen, and nitrogen. Within a few centimeter depth of soil, there are different amounts of oxygen, moisture, light, and nutrition, increasing the diversity of soil microorganisms. Now, as you know, different categories, different types of microorganisms require different environmental, chemical, physical features for their survival. Now, some might need higher levels of oxygen, some might need lower levels of oxygen. Similarly, in moisture, light, and nutrition, they require various conditions and various levels of these substances. So, when you go into several layers of soil, you can see many various different types of microorganisms surviving and living in these levels of soil. So, the top few centimeters of the soil consists the largest community of bacteria. Microbial number declines rapidly with increasing depth. Now, the reason is when you go into deeper layers of the soil, these contents such as oxygen, moisture, light, etc. Reduces drastically. Now, this reduces, so the number of microorganisms and the varieties of microorganisms in deeper layers is less due to the lack of these components. Majority of soil microfilariae, microflora, is represented by bacteria. In addition, there are fungi, algae, protozoa, and actinomycetes. Now, in any environment, in any ecosystem, the most commonly found microorganism is the bacteria. So here also they say majority of soil microflora is represented by bacteria. In addition, you can find these fungi, algae, protozoa, and actinomycetes like varieties as well. Actinomycetes, despite of being a member of domain bacteria, usually it is mentioned separately due to their significances. Now they consider it as a separate group, although it comes under domain bacteria due to its vast characteristics and unique features. These microorganisms play a major role in decomposition of complex organic substances and participate in cycling of elements in biogeochemical cycles. Now here these elements are oxidized and reduced by microorganisms for their metabolic requirements. Now regarding this, we'll be talking in detail in the slides to come. And here, as you know, cycling of elements in biogeochemical cycles is also important due to the reason of the lack of continuous supply of nutrients and elements in the environment. So whatever the available nutrients and elements need to be recycled within the environment so that it can be used again and again by the organisms and other living organisms found in the ecosystems. Now here you can see some images of fungi on the top left. These are some soil fungi. You can find these fungi, algae, and these protozoans and actinomycetes mainly in soil samples. And on the top left you have some such fungi, and on the top right you have some algae, and on the bottom left you have some ciliated protozoans. Now these have been obtained from a sample of soil. That's a microscopic image, and on the bottom right you can see some actinomycetes filaments, and this is how they appear when visualized. Now when you consider these microorganisms play a vast role in maintaining the soil context and the texture, and aids in many numerous ways to continue soil life and also life above soil. So we'll be talking about these methods of contribution one by one, and starting off first we have mineralization. Mineralization is the decomposition of plant and animal residue by using extracellular enzymes of bacteria and fungi. These enzymes break down complex organic materials into simple inorganic materials such as carbon dioxide and water. This is the major process by which plant nutrients are made available and also recycling. So now, as all of us know, mineralization is the breakdown of complex organic molecules into simpler forms using extracellular enzymes. Now, these extracellular enzymes are produced by the microorganisms and they are released outside of their body to the immediate surrounding environment, and these extracellular enzymes carry out the digestion process, and then the complex organic molecules are broken down into simple inorganic molecules such as carbon dioxide and water. Mineralization helps in following ways. The first one is it helps to remove plants and animal debris from the earth's surface, allowing other organisms to live. Now, all of us would be facing an issue if all the dead organisms, all dead plants and animals started accumulating on the face of the earth. There will be no space for the newly formed organisms, and we all will be facing issue with the space for living. So by the mineralization process, these dead organic matter, organic bodies of plants and animals are decomposed and they're removed from the face of the earth, and the nutrients included in them will be removed, and so then they can be absorbed by plants and animals and utilized by the animals. And the next point is recycle minerals which are found in limited quantities on the earth. Now, as I explained before, minerals are in limited supply, so they need to be recycled to be used over and over again. Moving on to the second point, the role of microorganisms in the carbon cycle. Now, the carbon cycle explains how carbon is being cycled through ecosystems and bodies of living matter. All organisms contain a large amount of carbon in organic compounds such as cellulose, starch, proteins and fat. Now, all of us must be aware that these compounds such as carbohydrates, proteins, lipids, they contain carbon as one of the major elements in them. So they contain a large amount of carbon in them. Photosynthesis is the important first step in carbon cycle, in which the atmospheric inorganic carbon dioxide is reduced, or you call it fixed, to form organic compounds by photosynthetic organisms. Photoautotrophs such as plants, cyanobacteria, algae, and photosynthetic bacteria fix carbon dioxide using energy from the sunlight. Now, these are the primary producers in an ecosystem. They consume this carbon dioxide, and with the help of chlorophyll pigments and sunlight, they carry out photosynthesis, and this carbon is taken into their bodies, and then the producers produce the food where the consumers then consume it, so that they could be the elements and carbon are incorporated into the bodies of the living organisms. Chemoheterotrophs such as animals and protozoa depend on organic compounds produced by autotrophs to utilize them as their carbon source. That's what I just explained. Now, through food chains, carbon fixed from carbon dioxide by autotrophs transferred from organisms at lower trophic levels to the organisms at higher trophic levels. Now, once the primary consumer consumes the primary producer, in a food chain there can be several trophic levels. For example, around 4 or 5 trophic levels can be found in a basic food chain. So once the primary consumer consumes the primary producer, progressively the second, third and fourth consumers will consume the organism from the lower trophic level. So the carbon will be incorporated into their bodies with this process. Both autotrophs and chemoheterotrophs release a part of their fixed carbon as carbon dioxide to the atmosphere through respiration. This carbon dioxide is again made available for autotrophs. Now, as all of us know, all living organisms need to respire. As a result of this process of respiration, carbon dioxide is released to the atmosphere in the form of gaseous carbon dioxide. This carbon dioxide can again be utilized by the producers, primary producers, to carry out their photosynthesis process. In chemoheterotrophs, undigested food is released to the environment as feces, which is later decomposed by soil microorganisms. This now part of the food or the organic matter taken into the bodies by consumers cannot be broken down or digested within the bodies of the organisms due to the lack of enzymes to digest those components. So this undigestible part will be released back to the environment through feces, and these feces will be decomposed by soil microorganisms, and the carbon and other elements and nutrients in them will be released back to the environment to be used by the plants and other species which consume them from the soil. Rest of the carbon fixed in organisms remain within them until they die. Once the organisms are dead, these organic compounds are decomposed and carbon dioxide is returned back to the atmosphere. Now, the part of the carbon which is not excreted and removed from the body in the form of feces will retain in the body and its structures. Now, as you know, carbon and other elements are required for the production of organelles and other components in the body. So they will be remaining in the body until the death of the organism. Once the organism dies, the microbes, the fungi, the bacteria and other microbial populations present in the soil will carry out digestion, and they will carry out decomposition and release the nutrients and minerals into the soil, which can be utilized by the producers. Microorganisms, mainly bacteria and fungi, play a major role in organic matter decomposition. Now, bacteria and fungi carry out decomposition of this organic matter, and they are the main decomposers found in the environment. Microorganisms play another major role in carbon cycle in relation to methane gas. Ocean sediments contain a large amount of methane. However, about 80% of methane generated within ocean is consumed by microorganisms called methanotrophs before it reaches the atmosphere. Now, methane is CH4. CH4 consists of a carbon, and here too this needs mentioning in the carbon cycle, because 80% of methane gas is generated within this ocean, consumed by microorganisms such as methanotrophs. This needs to be included in the carbon cycle. In addition, now this is an aquatic example. In paddy fields, methanococcus like microorganisms involved in production of methane. That's a terrestrial example. Then moving on, despite of the above, methanogenic bacteria in the ocean's depth are constantly producing more methane. This methane production continues throughout. Now this is a simplified diagram of the carbon cycle. Now here you can see the at the top the carbon dioxide in the atmosphere. Now atmosphere consists of carbon dioxide in the form of gaseous carbon dioxide, and here on the right hand side you can see the sea, and this ocean, this consists of dissolved carbon dioxide. Now both these forms of carbon dioxide are taken up by the primary producers. On terrestrial environments, they are the plants, as shown here, they are the plants and trees and other microorganisms such as cyanobacteria, etc. And in the aquatic environments, the aquatic plants and aquatic microorganisms utilize this carbon dioxide. As a result, they carry out photosynthesis, and this carbon is incorporated into complex molecules, and once the organisms living in these terrestrial and aquatic environments consume these primary producers, the carbon would be incorporated into their bodies, and as the trophic food chain continues, the higher levels in the food chain will obtain the carbon by consuming the organisms from the lower trophic levels. In addition, you can see there's an eruption of the volcano here. Now through this eruption of volcanoes, carbon present will be released in the form of carbon dioxide due to the process of combustion. Here carbon dioxide gas is again added to the atmosphere. Now once the living organic matter such as all the plants and animals are dead, they get added to the soil, and here on the bottom you can see the microbial respiration and decomposition. The microbes carry out decomposition of this dead organic matter and releasing the carbon dioxide through the respiration process back to the environment. Now there's a human interference mentioned here, which is the human emissions. For example, automobile fume emissions. Now they might contain carbon dioxide, and in addition industrial carbon dioxide emission. Now this can actually be a drawback. Now the main reason is carbon dioxide is one of the main greenhouse gases, and with the excessive increasing levels of the carbon dioxide in the environment, global warming can take place, and many adverse effects can be resulted due to this. So this is the summary of the carbon cycle. It's a simplified diagrammatic representation. So moving on, the third point, role of microorganisms in the nitrogen cycle. Similar to the carbon cycle, nitrogen also has a cycle where the nitrogen is being circulated through ecosystem. All organisms require nitrogen to synthesize protein, nucleic acid, and other nitrogen containing compounds. About 80% of molecular nitrogen available in the atmosphere. Now 80% of the content is molecular nitrogen, or N2, which is available in the atmosphere. This is the main reserve of N2, which is the atmosphere. Here nitrogen is in the gaseous form. This is not biologically available for organisms. Therefore, it is essential to convert that atmospheric molecular nitrogen into bioavailable forms of nitrogen. And now, what are bioavailable forms? Bioavailable forms are the forms of nitrogen that can be utilized by plants and other primary producers. Now, examples for bioavailable forms are in the form of ammonium ions and nitrate, or NO3- ions. Certain groups of microorganisms are able to fix gaseous molecular nitrogen into bioavailable forms of nitrogen, such as ammonia, nitrate, and nitrate. I just mentioned that. Therefore, nitrogen available on the earth, organisms, and the atmosphere must follow cyclic paths. There are 4 key steps in the nitrogen cycle. They are ammonification, nitrification, denitrification, and nitrogen fixation. Now, once you read these 4 steps, the names kind of sound similar, but these are 4 distinctly different steps, and each step mentioned here have their own individual functions. So let's look into each of these steps one by one. Starting off with ammonification. More than 90% of organic nitrogen in the soil exists as proteins. Proteins from dead plants and animals are decomposed by extracellular proteolytic enzymes secreted by microorganisms into amino acids. Now, you might be aware that proteins are made up of small units known as the amino acids. This is the basic structural unit of the proteins. Sorry. These amino acids consist of an amino group, which is the NH2 group. Now, these microorganisms carry out extracellular digestion and release this NH2 amino acids from the complex protein molecules. The resulting amino acids are taken into microbial cells and are then subjected to ammonification, in which amino groups of amino acids are converted into ammonia. Now, the activity of these microorganisms which carry ammonification is the conversion of the amino group, or the NH2 group, into ammonia. In moist soil, ammonia is solubilized in water to form ammonium ions. These ammonium ions are utilized by plants and soil microorganisms. Ammonia in dry soil rapidly disappear into the environment. Now, once this ammonia is produced, if water is available in the environment, it dissolves quickly due to its high solubility. So, once ammonia dissolves, it forms the ammonium ions. So, this ammonium ion can easily be absorbed by the plants and other primary producers. Now, if by any chance, if the environment which the ammonia produced is dry, the ammonia gas will be released to the atmosphere before long. The next step is nitrification. Nitrification is the process of oxidation of nitrogen into the ammonium ion to produce nitrate. This process is done by nitrifying bacteria living in 2 stages. Now, this nitrification process consists of 2 stages, 2 distinct stages, and there are two types of microorganisms that carry out these 2 steps. In the first stage, microorganisms such as Nitrosomonas oxidizes ammonium ions into nitrites. Now, here you can see this conversion. Ammonium ions are oxidized to nitrite. The nitrogen in ammonia is oxidized to the nitrogen in nitrite. This conversion is carried out by Nitrosomonas and Nitrococcus-like microorganisms. In the second stage, microorganisms such as Nitrobacter oxidizes nitrites into nitrates. Now, here the further oxidation of the nitrogen nitrite occurs, producing nitrate. This conversion is carried out by the Nitrobacter microorganism. These are scientific terms and they should be italicized when typed and underlined when handwritten. Plants utilize nitrate as the source of nitrogen. Therefore, microorganisms play essential roles in providing nitrogen in bioavailable forms for plants and animals. Now, the forms which can be absorbed by plants is in the form of either ammonium ions or nitrate ions. Nitrite or NO2- ions cannot be absorbed directly by plants. The next step is the denitrification process. In the absence of atmospheric oxygen, some microorganisms use nitrate and reduces it into N2. This process is called denitrification. Denitrification causes loss of nitrogen into the atmosphere. Therefore, it reduces availability of nitrogen in the soil. Now, this denitrification process really reduces the availability of nitrogen in the soil because the nitrate in the soil is converted into N2. N2 is a gaseous form, and due to this gaseous nature, it would be released to the environment. Therefore, it reduces availability of nitrogen in the soil. So, nitrogen reduces, reducing the availability of it for the plants. Denitrification frequently occurs in waterlogged soils where limited oxygen is present. Pseudomonas species converts nitrate ions into molecular nitrogen through the following steps. First, the nitrate goes into nitrite. Then the nitrite is converted into the nitrous oxide, which is N2O, and then the nitrous oxide is converted into nitrogen gas. Now, this is the process where nitrate ions are converted to nitrogen gas. The nitrogen gas is released to the atmosphere, reducing the nitrogen availability in the soil to be utilized by the plants. The next step is the nitrogen fixation. The process of conversion of nitrogen gas to ammonia is termed as the nitrogen fixation. Now, nitrogen fixation is making the atmospheric, in simple terms, making the atmospheric nitrogen available for the plants to be absorbed in forms which facilitates its absorption. Bacteria that are able to fix nitrogen require nitrogenase enzyme. Nitrogenase enzyme is inactivated by oxygen. Now, in a previous video, you might have learned that nitrogenase is an enzyme which facilitates the nitrogen fixation. And if oxygen is present, oxygen hinders and prevents the efficient functioning of this nitrogenase enzyme. There are two types of nitrogen fixing microorganisms. They are free living and symbiotic. So, starting off with the free living nitrogen fixing bacteria, they are abundantly found in the rhizosphere. Now, what is the rhizosphere? Rhizosphere is the soil immediately surrounding the plant root. It's the soil that is in contact or just in the close vicinity of the plant root. For example, Azotobacter species. Many cyanobacteria are able to fix nitrogen and make them available for the plants. For example, Nostoc. Nostoc is a cyanobacterium which is able to fix nitrogen freely without interacting with any other species of organisms. That is the definition for free living. They don't need the assistance of any other species or organism to carry out this nitrogen fixation process. These organisms exhibit mechanisms to prevent the exposure of nitrogenase enzyme to the atmospheric oxygen. For example, Azotobacter species have a high rate of aerobic respiration. Now, when there is a high rate of aerobic respiration, the rate of utilization of oxygen is really high. So, the availability of oxygen in the immediate environment of these microorganisms the environment is very less. So, due to this less availability of oxygen, the nitrogenase enzyme can function effectively as an environment which is very similar and close to the anaerobic environment is created. And the next cyanobacteria consists of heterocyst. Now, in a previous video, you might have seen the heterocyst is a special kind of cell found in some cyanobacteria. These heterocysts consist of thick walls. Due to the availability of these thick walls, oxygen in the environment cannot penetrate and reach the nitrogenase enzyme which is found within the heterocyst. So, here the nitrogenase enzyme can carry out its functioning independently without being hindered by the oxygen in the environment. Some anaerobic bacteria such as Clostridium species also fixes nitrogen. Now, an additional fact that you must be aware is all Clostridium species are anaerobic kind of bacteria, and some species also involved in the free living nitrogen fixation process. Now here you can see an image of nostoc. This is what it looks like in real life. This is a cyanobacterium which is able to fix nitrogen freely. Then moving on, symbiotic nitrogen fixing microorganisms. They play an important role in agricultural crops such as leguminous crops, which are soybeans, beans, peas and peanuts. These symbiotic nitrogen fixing bacteria are commonly known as rhizobia. Leguminous plants are specially adapted to facilitate symbiotic nitrogen fixation. Plants form root nodules where anaerobic conditions and nutrients for bacteria are given. In these root nodules, which are produced by the leguminous plants, anaerobic conditions are produced, so the nitrogen fixation process is facilitated, and also in return nutrients for bacteria are given by the plants. Bacteria fixes nitrogen and makes it bioavailable to be used by the plant. Some non-leguminous plants can also fix nitrogen symbiotically with different microbial combinations. Now here lichen, a combination of fungus and an algae or cyanobacteria, also fixes nitrogen. Now here you can see the algae and cyanobacteria have photosynthetic ability. Now they carry out photosynthesis, and as a result the fungus and algae or cyanobacteria combination is able to fix nitrogen, and this is a symbiotic mutualistic kind of interaction where both parties are benefited. In rice paddy waters, a small free floating fern which is known as azolla fixes nitrogen symbiotically with Anabaena species. Now here you can see some images. On the left side you can see the leguminous root nodules. Now these are found in legume roots, and they facilitate and provide anaerobic conditions for the symbiotic nitrogen fixation. And on the right side you can see the free floating fern which is azolla, and this along with Anabaena involves in symbiotic nitrogen fixation. So moving on, interactions of soil microorganisms relevant to plant growth. Microorganisms in the soil directly interact with the plants. Now they are directly in contact with the plants, so there is a direct interaction between them. Interactions in the soil such as rhizosphere, mycorrhizae and endophytes with symbiotic relationship of bacteria, fungi and plant cells. Those are the interactions. These microorganisms are beneficial to the plants in numerous ways, such as nitrogen fixation, increased uptake of water and nutrients such as phosphorus, secretion of plant hormones such as indole acetic acid, increases iron uptake at iron limiting conditions, and defense against pathogens. Now here these microorganisms are beneficial to plants in numerous ways such as nitrogen fixation. That's the example which we discussed before. And they also increase uptake of water and nutrients such as phosphorus. For example, mycorrhizae mainly involves in facilitating the uptake of phosphorus element by the plants. When the mycorrhizae are available in association with the plant roots, it enhances the absorption of phosphorus by the plant. Then next, secretion of plant hormones such as indole acetic acid. Indole acetic acid, or in short you call it IAA. This is a plant growth substance or a plant growth hormone coming under the category of auxins. IAA is an auxin, and this promotes plant growth, increases iron uptake at iron limiting conditions, and also they provide some defense against pathogens. You can find pathogens because the soil environment and a large variety of microorganisms can be found, and some of it might be pathogenic. In return, plants provide organic compounds essential for the microorganisms. So microorganisms also require certain nutrients and components that are essential for their survival. These are provided by the plants in form of organic compounds. Then moving on to rhizosphere. This is a kind of symbiotic interaction between plant roots and soil surrounding the root surface for about a few millimeters. Now this is the immediate soil environment of the plant that is in contact or interaction, that interacts with the root of the plant directly. This microecological zone is called rhizosphere. Rhizosphere is considered as the most diverse and dynamic habitat on Earth. Now that point is really important. Rhizosphere is considered as the most biodiverse and dynamic habitat on Earth. Now what is the meaning of most biodiverse? Most biodiverse in the sense it consists of a large variety of organisms within the rhizosphere. So they call it the most biodiverse ecosystem, and it's a dynamic habitat. Now what's the meaning of dynamic habitat? There is a constant moving of nutrients and other minerals, etc. Through this rhizosphere, because there is exchange of these substances with the soil and the plant roots, and this is a very rich system. So that's why they call it a most biodiverse and dynamic habitat on Earth. That's an important point. Microorganisms in the rhizosphere feed on the compounds exuded by roots such as sugars, amino acids, and various aromatic compounds. Microorganisms compete, antagonize using antimicrobial compounds with each other for resources such as nutrients, space, and water in the rhizosphere. Now here you can, because the rhizosphere is a very small area, there can be competition for nutrients, space, and water, which are essential for the growth of all organisms. Bacteria are the most numerous organisms in the rhizosphere. Not in the rhizosphere only, in any environment, bacteria are the most numerous organisms. Three most common genera of bacteria inhabiting rhizosphere are Pseudomonas, Bacillus, and Agrobacterium. These are the three main examples which inhabit the rhizosphere. They are most commonly found in the rhizosphere. Root exudates act as chemical signals for bacteria to move towards the root surface. Now this root consists of several chemical secretions, and these chemical secretions act as chemical signals for the bacteria, and they respond to these chemical signals and move towards the root surface. Both pathogenic and symbiotic fungi associate with the rhizosphere. Then moving on, mycorrhizae. Now this mycorrhizae consists of two components in the name itself. First is myco and second is rhizae. Myco means fungus, rhiza is the root. So this means fungus associating the plant root. Mycorrhizae are symbiotic associations between plant roots with fungi, as its name suggests. Almost all land plants have symbiotic association with one or more mycorrhizal fungi. Mycorrhizal fungi extend the surface area over with nutrients, and water can be uptaken by the plant. They can reach small pores in soil where plant roots cannot reach and uptake nutrients. So this mycorrhizae can grow into spaces through the small spaces present between the soil particles which the plant roots cannot reach. So once this mycorrhizal growth occurs, they involve in absorbing the nutrients and water and making them available for the plants. So the areas which cannot be reached by the plant roots will be reached by this mycorrhizae, and as in return the plants will obtain the water and nutrients through them. Most significantly, mycorrhizae increase uptake of immobile nutrients such as phosphorus, zinc, and copper. What are immobile nutrients? Immobile nutrients are nutrients which are not transferred or transported from mature regions of the plant to the younger regions. These are not transported, that's why they are known as immobile nutrients. The examples are phosphorus, zinc, and copper. These elements are made available to the plant by the action of mycorrhizae. In return, mycorrhizae receive organic carbon from the plant. Now that's a mutualistic relationship. So as a result, mycorrhizae can obtain the organic components, nutrients provided by the plant for its survival. Now here you can see an image of mycorrhizal growth. These white patches which forms a net-like appearance are the mycorrhizae, and they facilitate and help the uptake of nutrients and water by the plant. Role of soil microorganisms in improving soil quality. Free-living soil microorganisms and those associated with root surfaces play a major role in improving soil quality. Microorganisms are involved in the formation of stable soil aggregates, which are characteristics of good soil structure in fertile soils. Fungal filaments, actinomycetes filaments, and polysaccharide gums or slimes produced by bacteria are involved in soil aggregation formation. Now, in the formation of these such stable soil aggregates, the fungal filaments, the actinomycetes filaments, and the polysaccharide gums and slimes play a major role. Now, these filaments and the gums are the main structures which hold the soil in aggregation. So once these aggregates are formed, there can be spaces formed for the gases to be stored between the soil particles. So these gases can be utilized by the plants and uptaken by the roots. So such stable soil aggregate containing soils are very suitable for the growth of crops and other growth of crops as well as other plants. So here you can see an image of stable soil aggregates. This is how what a good quality soil would look like. These small aggregates you can see are the stable soil aggregates formed due to the action of fungal filaments, actinomycetes filaments, and the polysaccharide gums. These polysaccharide gums are sticky substances, and due to this texture, these aggregates will be easily formed. So thank you very much for watching this video, and if you learned something productive from this video and you enjoyed it, you may subscribe our YouTube channel and click the bell icon and share this YouTube channel among your friends so that something productive will be added to your lives, and also this would help you to face your LL examination successfully. Good luck. Thank you very much.